Hi. In this video, I am going to go through and I am going to talk you through our pipeline. Now, a couple of things to point out. The pipeline for this project currently has a bunch of information censored. I'm going to talk you through it, and so you can come in and you can review this if you're really just getting stuck during the annotation activity. However, the idea is more that you're developing a sense for this pipeline, that you're coming to understand it. And one of the bigger ways, better ways to do that, because let's face it, this is not short. It's a very long script file, and it's going to take you a while to read through, let alone come to understand, is really leaning into kind of hierarchies of scale. That I have done a lot of high level summaries of what's going on. These are always going to be indicated by multiple comment symbols that are present on either side. That's what allows me to structure these. That's what gives them in the hierarchy. So the more comments I put up here, the further to the right it gets indented. And so we can see things like this README section. That's describing that, hey, you probably shouldn't be changing most of this. Like, this is pretty much intended to be run as is. I want you to understand how it's used, but the actual mechanics of it are often pretty tricky. I'm going to talk you through some of them and help you try to understand what's happening, but that's the idea. So do keep this up. I'm just going to push this over to the side of my screen, make this a little bit bigger so that on y'all's laptops you're not struggling too much to read what's happening. Okay, so with that, actually let me give my little face, some extra room, and that was weird. All right, moving forward. I'm up in the section 1, clearly. The section 1 is about setting the options and the environment. It's not really stuff that you should be changing. I do use a really cool strategy where I store a bunch of this information in opt, and I'm going to show you what that looks like. But that's really the point of this first section, is to set up an object that's going to be storing a lot of the different objects and data sets made throughout this pipeline that aren't directly related to the actual results. So this is going to be more the options that are controlling things. So the first step is to use this list function. And you'll notice the list function currently, no arguments. And that's because I'm not storing anything inside the list right now. I'm simply creating a list class object to actually then store stuff in. Here is where I'm actually starting to store stuff in it. So the first thing I do is I extract a container. However, that container is initially null. It doesn't have anything in it. It's not been created. I'm going to assign it data. And here's the data I'm going to assign it. Run all together. It stores that value inside of an object called WD. And if you're being clever, you might be able to figure out what WD is going to stand for. It's eventually going to be going right here. Next up, after having set that, we're going to get a couple of pieces of information. So the first thing I'm going to get is this rstring version. That's all of this text down here. That's not the most useful thing. However, if I run my gsub function and I correctly add in parentheses, actually I guess I could fix that. There we go. First change, made it to line 22 before having to tinker. This is always the problem with this video. With that, I've stripped out everything that matched with a space followed by a literal open parentheses followed by any character repeated 0 or more times. That is a match of all of that space, literal, open parentheses, and then a bunch of text. I then replaced it with nothing, stripping that text and leaving me with just our version 4.5. I then remove these spaces because I don't know how much y'all have worked with this, but it does turn out that spaces often cause problems in file names. Not necessarily because your computer can't handle them, but because it's hard to reference spaces because they're white space. You can't see them. And so it's nicer often to remove them and replace them with something that is present. Finally, I then assigned it to a previously empty container, creating that container and storing the value inside of it. That value clearly representing the version of my R shown right here, and that's the same value. Next up, I am using a function called file path. It is a function that creates file paths. It's starting with a location in my current directory. That's what I've currently highlighted. Those symbols together stand for current directory. Then R packages, and then that name. Run together. On my computer, it looks like this, but if you're running this on a different computer, say the Linux server or a Mac computer, the text might be slightly different. However, that's the point of the file path, is it helps make the file path regardless of differences between computers. That said, most computers are pretty flexible these days with how the file path can be written. There's a lot of exceptions to that, but that's often true. Next up, I am using a path that has a little bit of context we really got to think about. This path is only useful present on the EduPod. So store, scratch, the name of our class, followed by my initials, then shared R, and then R26A Spring. So this is actually a directory I created and spent a lot of time setting up because it takes a while to install all this software. Now, this is obviously only present if you're working on the EduPod, and that's why this text is here. Next up, I am combining several different values. If you don't recognize these are each different packages. These are packages that can be installed from a, well, it's called a repository, but a collection of packages known as the CRAN, the Comprehensive R Archive Network. Dang, I pulled that right out. It's the common place when you're using like install.packages that R installs from. Now these packages, some of which are actually also on the CRAN, are not going to be installed from it. Instead, we are going to be installing from a separate collection called the Bioconductor suite of packages or Bioconductor repository or whatever they call themselves. But this Bioconductor collection is a bunch of biology related packages. So I'm just going to run those lines of code before I forget to run them. And then finally, I'm going to go down here and I can see that there's a bunch of text. It really mostly matters to Windows users. There's a program you might need to install depending upon like the hardware that you're using. There's not an easy way for me to predict this, and just basically if you run this a couple of times and it fails, you may need to install this if you're on Windows. If you're not on Windows and it still fails, there might be something else going on, and frankly, from my experience, it's just not worth it. The EduPod's working pretty well this semester, so just move on over to the EduPod. One thing to watch out for, though, are these boxes that are going to ask, do I want to install from a source requiring compilation? That can be great if you have compilation tools installed and if you have a bunch of free time, because they do take a while. Typically, I suggest you say no to those. If it still doesn't work, then that's when maybe you can run it again and say yes and actually compile the newer version for your computer. Compiling, just for the record, is this process of interpreting the code in a way that your computer knows how to handle it. It requires kind of translating between the human code that we write, so this is all intended to be read by humans, and transforming it into a way that the computer can understand better. So I'm just going to run it. It's going to do a bunch of things in the background. I'll talk you through a couple of them. So this is my newest version. I kind of update this whenever I make changes to this configuration package, or configuration script file actually. It's saying that it's checking if the opt object had all of that stuff that we should have been putting into it. If not, it's going to tell you to go put some stuff into it. It's then setting the working directory based on that WD container that we created, and there we go. We can see that just went through. It then downloaded a copy of this script to that working directory. That's just so that if you wanted to, you could go in and take a look at what I've done. Just if I go to the working directory, I can see it right there, project shared functions. So that's actually what's being run currently on the computer is project shared functions. And it just made a copy of itself just for your records. Determining if the shared package directory should be used. And so it tried to see, could it access this directory? It turns out no. And so it's ignoring that setting. It's not going to use that shared directory because it doesn't have it. It's not on the EduPod right now. This is my personal computer. That's expected for a personal computer, but if you got this on the EduPod, that would have been unusual. It does seem to be working on the EduPod right now. I then set what's known as the lib paths, and for whatever reason they put a dot in front of that. I think it's a capital P, yeah. The lib paths are the locations where R is going to look for packages. So the first place it's going to look is this new directory that I've created with my R number, and it's nested inside the project, and that's going to make it very easy to control the version of software because it's going to look here first. And if it doesn't find a package, then it's going to look here. But depending on how your computer's set up, you may not have permissions to change that library. That's why we made a separate library. So not only does this allow us to easily control what version's being installed, it also allows us more broadly to be able to install despite the differences between how our computers are arranged. It then installed those packages. Mine had already been installed, so it just says you're good, and then it displayed configuration successful. So I did say that I expect some students to have issues here. Sometimes just running this a couple of times as you kind of install some of the needed packages, the prerequisite packages for installing other packages, it kind of resolves itself. And sometimes you can run it multiple times and it doesn't change anything. In that situation, go through these debug steps, but maybe the best option is just to move on over to the EduPod. This project's hard enough without you struggling with the packages. So finally, it then checks to make sure like kind of the essential and the really hard package to install is able to be loaded. This is in fact the package that's currently broken. Let's see what version I'm installing. So we're installing well, not the worst version. Unfortunately, this one is still not working, and so this code maybe not really. Actually, in fact, this code might be what I change as I release the next version of this script file. Hopefully fixing it after I finish this video. Section 2. All right, so in section 2, we are loading sample sheets and checking the quality. Okay. So the sample sheets for me have been uploaded to a Box repository, and I can easily download them, which is just really handy for sharing this. It's going to download from these two different direct Box links, and you can tell they're direct because they end in the extension of the file and not like .html. They save them to these two different files, and those are going to go into my working directory. So if I refresh this over here, I can now see that they've appeared. The files are then read using a read.delim function. I was kind of sneaky and I made y'all convert your files into CSVs because I'm a jerk, which means you're gonna have to change this. This is not gonna be the right code for reading your data into R. You're gonna have to pretty much just rewrite this. In fact, what I recommend is you just go through here, comment out my old code, and then start writing your own version of group 1. Read dot, I don't know, depends on what the file is. You'll figure it out though. Remember, you do have that import dataset tool to help you figure out that code if you are actually struggling with it. Although I do hope maybe you remember at this point how that works. Regardless of how you do it, we then need to check to make sure you did it right. And so the next section is going to go through and it's going to start checking for various issues. And really it's only using functions that we use in this class. I expect you should be able to understand all of these. So for example, this determines the class of group 1. It then is able to resolve that % in %, and that's going to say true or false is data frame of value inside the class of the object. It's then going to negate that. So if the data frame was not a value inside the group 1 object, you get this stop message that says, oh no, it's not a data frame. Well, group 1 not found. It's because I didn't run this code. There we go. Maybe I should have added a code for that. It is a data frame though, so that's not going to be a problem. Obviously, if I changed group 1 to like a vector, so here I've changed it to the vector of letters. If I try to run this, oh no, it's not a data frame. And that just tells me I need to go fix my reading code. Anyways, this is going to repeat for group 2. Next up, we've got some values being combined into a new container. So that's just storing a character vector inside of OPT checked columns. I then check each of those values. Are they in the column names of group 1? Those are all in those column names because this is what the column names of group 1 look like. And then I summarize that since a if statement requires a single logical value, and this is saying, yes, they are all true. Had any of them been false, so like if, one of those hadn't been working, I would have gotten an error message. But that's not what happened, so they worked good. Next up, it's checking the number of unique values and measuring the length of that. It's then checking the number of rows, and it's saying, is one less than the other? If it was, then that would mean that you had some duplicated sample IDs, either in group 1, group 2, or in the case ID columns. Usually these are not intentional and they should be removed. There are some reasons why you maybe could have duplicates, and in fact there is something here that's not currently being tested that I probably should actually add, and that would be to combine these group 1 and group 2 and see if there's any duplicates in there. That could also be a problem. But right now, that's all that's testing for. Just make sure you don't have any duplicated case IDs or sample IDs. It can cause problems later on, as well as it's just not good experimental design because you're biasing it, your analysis towards one person overly much. Okay, next up, I am checking the total number of rows added together across both groups. And then I'm comparing it to the number 250. If you do have greater than 250, it's enough that you might start running into issues, either just because you're downloading so much data or because the amount of memory needed to store the information in your computer starts to run into other issues with your computer's hardware. I generally just recommend don't push it. This is just a class project. If you really needed to, you could get access to a nicer computer, just use the EduPod, which is a nicer computer than yours at least. You can't make a laptop with as much storage as the EduPod has, so that's not a bold claim or anything. Maybe if you're from the future, maybe that's untrue. But then it tests, is it greater than 100? And it's like, this one is strongly considered. You're probably going to run into some issues. And then this one saying, if any of your groups are greater than 100, at least one of your groups is pretty big, and maybe you should consider it. Neither one of these are absolutes. There are reasons why you may want to have so many cases, but there's actually kind of a lot of stuff that can go wrong as you start to get these larger groups. Honestly, it's just kind of interesting to make you have to think about this, and you should be thinking about this. Yeah, I'm going to move forward. Next up, considering how poorly controlled these experiments are. So each case is a different patient. That's a lot of variation. There's little expectation of statistical reliability at small case numbers. We needed a large number of cases. Frankly, something that you probably don't have the context for is these are crazy experiments that we're doing. Differential expression analyses are historically extremely expensive. It's a very bespoke situation, you're focusing on a specific time point, And you're often having to sequence very, very similar things. That's a lot of money that's targeting something very specific. And the reason we can do that with these projects, well, first of all, is that they're purely exploratory. We're not trying to like discover extremely fine level biology. We're simply looking for correlations that might be interesting and worth further study. But the other thing is that this data has been shared and was collected in mass from a very large grant with a lot of the costs kind of maybe not amortized, but defrayed across multiple different systems. So I mean, there's just a lot of money that's been put into this that's not typical of these types of experiments. But anyways, we can have large case sizes, and because of all this variation that we know, we should have larger case sizes. If your case number ever drops below 6, I need you to talk to me. As we get deeper into the project, I will sometimes allow you to keep going if you drop down to like 5, but it really depends on your project and really why you drop down below 6. So you need to reach out or just redesign your experiment, one of the two. Next up, it is checking if the type of data, so let's just highlight that and show what this looks like. Gene expression data, are they all gene expression data? Yes, they are. That's fantastic. We wanted that to be true. It can only use gene expression data. So if they are, not all gene expression data, ran it all together, then it would have produced a stop message. But as we can see right here, that's not true. So this doesn't happen, and we continue through. And we now have reached section 3, search for data to download. You know, this is actually not helping, so I'm going to close this down. You should keep it open. This will help you navigate things, but I just kind of need some more space as we get into this larger code. All right, next up, we got search for data to download. This is an interesting step. I think students get confused because they expect things to work very nicely and cleanly, but really what this is doing, it's working around the limitation of the system. Those sample sheets do not have the needed information to restrict the download step that's going to happen later on. Unfortunately, to get the values needed to control that download step, I'm going to need to actually first search for the data. So what I do is I load the package just in case you were skimming through and you hadn't loaded the package yet. This is more of a safety. We should have already done this. I then detect what are the unique project values, just in case you had multiple project values, and I store that inside of an object. This is just right now from my project, the colorectal cancer data set. And here is this big step where I am searching for essentially all cases associated with this project. Now, this is going to take a second, so I'll talk while it's thinking here. But what it's doing is it's going through the GDC. It's isolating a particular project, in this case, the TCGA-COAD. It is then isolating the transcriptome profiling data of type gene expression quantification. So there's other ways of doing transcriptome profiling. You could look at small RNAs, you could look at gene editing. There's a lot of different analyses you might end up doing, but we wanted gene expression quantification. Specifically, we wanted an algorithm called star counts, which is kind of the input to our final analysis later on. So we specifically wanted star counts. I believe that's nowadays the only data available of gene expression quantification. Obviously, we only have access in this class to the open access data. We would have had to request special permission to get closed access or controlled access data. But this is now completed. And if I print it off, we can see that it's not very useful. It's not a very useful object. If we try to investigate what it is, we could say all right, well, what's this class of an object? It looks weird. It's a data frame. It's like a one-row data frame. That's weird. Well, we could open up the $sign and you could scroll through here, and really they're not all that interesting. Although one thing you may not know that I know is the as-is class can kind of store a lot of different stuff. So inside of here, it turns out that, sorry, I forget, I actually can't do that. An as is, I believe, is a class of list type. Let's see if that's true. Yes. And that means that we can open up its layers, or we can look at its length. Okay, it's a length one list type object. So what if we just look inside of it? What is that? Okay, there's a data frame inside. This is literally what I did way back in the day when I was trying to figure out what the heck is going on with these results. And it turns out that the way they store all this information is as a data frame stored inside of an as-is object, which is a class of list type, which is stored inside of a data frame, which is what query one is. Why? Don't know. In fact, it really doesn't even work well if you have multiple searches and you end up with multiple rows in that initial data frame. It's a very strange object. But we're just going to move out that data frame because we can work with data frames. Data frames should be fairly simple to work with. So I extract that data frame, having investigated it, saved it to an object called samdf, and that's going to be the data frame we use for a bit now. Complicated, right? But it's mostly just limitations. That's what a lot of this next section of code is dealing with, is limitations in the system. Okay, here we've got the sample submitter IDs. These are the IDs of the patients in combination with the sample. So it's the sample and the submitter's ID. So as we had different samples get submitted, they get different IDs, and so we can sometimes see, if I sorted these, I bet we could see some ones where it's the same patient. I'm just looking for the same numbers. It's not terribly common in this COAD data set, it looks like. It's fine, this isn't that important. I just wanted to make a point, but I think I'm just gonna skip ahead. Yeah, don't worry about it, it's fine. I'm looking to see, does it overlap our group 1 ID? So our group 1 ID, a much smaller, because it's just a subset of these 524 possible cases. I'm just looking for these 16, 17, 18, 19, 19 out of these 524. So if I run that code, it's going to tell me true or false, true, 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 so on. Are they in those sample IDs? I then save that as a new column, which has been added to the end, which is saying true or false, is it inside of our group 1? I repeat that for group 2. And so now I have 2 columns of true and false at the end. We can actually see some of our group 2 samples now are present in that section. And then finally, I create a third round of logic, which is combining those previous 2s and saying, are either one of these previous 2 true? That's going to be useful because we can use it to do things like reorganize our data or subset the data. So I could click on this and it's going to let me view that. It's going to currently be sorted based on that sample logic. So I put all my sample logics at the top. And then if I scroll down, I can see here are all the falses. But these are the cases that I want to use, and that's great. So what I then do is I take the sample submitter ID. This is the value I actually need, and I extract the it using sample logic. I create init barcodes using that. This allows me to have double checked all my work, make sure it's all being present in my search result, and then I can pull this out here. So you should have a couple of questions. So for example, one question could have been, couldn't we just have copied this sample ID and this sample ID and combine those to create this? And the answer is absolutely. However, this allows us to check to make sure that our sample IDs were in fact coming out in that analysis. This was more of a check than anything else. But if I then run this, guess I forgot I could talk during this time. If I run this, it's now going to be limiting the barcodes that it uses to just those values. I do want to point out that this system uses the term barcode like willy-nilly. It's used all over the place for different values. It's very frustrating. I don't know why. That's just what they chose to do. Don't think about it too hard. Next up, we want to just check these results. This is actually a section that is checking both the grouping information and the query results. So your group and query results, let's just say it's both. So the first thing is I'm just going to check to make sure, well, you did actually have data in both group 1 and group 2, right? Cool, those should be more than 0, and they are. Okay, did they overlap any? No, good. What about the other direction? No, that's good. So neither group 1 overlapped with group 2, nor group 2 overlapped with group 1. That's what we want. I then do that same process where I kind of do some weird subsetting of the query 2 object to pull out the sample df of the results. And this is just making sure that, these numbers all make sense. So this was 19, this was 54, this is 73, which is 19 plus 64. That all makes sense. I actually ended up with one of each of my groups. I then check to make sure that's true. So I just did it using math, but we can also use this logic. So are each of these values in these values? So some are true. For the ones that are not true, so the ones that are not true, they should be true in the other one. That's what this is asking. If it's in this one or in this one, and then saying, are they all true? If you had any falses here, somehow that would have indicated that somehow you ended up with barcodes that were in neither group. And that's usually an issue, something to do with this right here. Usually, honestly, a student having messed this up somehow. It happens. Or I could have messed it up too. I'm a student of the world these days. So it's weird to do these. I should edit that out, but I'm not going to bother. So there you go. You just get to catch me doing really weird stuff. Anyways, next up, this is checking to see are any of them duplicated. No, that really causes some sincere issues later on. This is actually a different way of doing that unique code that we did earlier. It's the duplicated function. The duplicated function, any duplicated values would have appeared as true. So if I really wanted to just demonstrate that, I could add in a duplicated value. So we'll just make this be, so we just added like the second value on twice to the end of it. So there they are. And so if I run duplicated, those last two duplicated values will return true. I needed an extra parenthesis. So so you can see that they're duplicated. Now, interestingly, the first one turns out to be false, and there's a lot of reasons for that. But in the end, this was just checking to see, are they duplicated? And then it summarizes it using an any function. So if any of them had returned true, which before my modifications, none of them did, it returns false because none of them were true. And finally, if you did get a duplicated value, this is just here to show like maybe why. This is the most common reason is that people have both normal and primary tumors here. So that's by far the most common reason. All right, next up we've got, well, you know what? Let's do one more thing. Not just that, but actually, you know what? No, we're good. Cutting it there. Don't make it longer, Nolan. That's right. I learn over time, slowly. All right, so we're now on section 4 out of 6, right? Yes, 4 out of 6. That's good. This is actually the step that's got the error in it, so I'll show you what that looks like, although the nice thing is it is still downloading the right data. So at 4, we are obtaining and structuring the data. We're not really doing a lot of analysis, but we will have to check some of the quality. So the first thing is it's going to download. Nice thing is I've already done this, so I don't have to wait for it. It's set up to download in chunks of 10. Please don't download an insane amount of data. Not only can you fill up the EduPod, your allotment on the EduPod at least. Yeah, you could fill up your I can't fill up my allotment. I have more of an allotment than you. That's what it is. You're fine. I have this in here though, just to keep you well away from storage limits on the EduPod, which I'd have to go look up. I think it might be 5. I think. I don't want to commit to that. Anyways, here's where we're telling it what to search for and download. Yeah, that's it. The API method, there's some other methods. The API one works the best and most reliably. If it does fail, that's what I should say. If it does fail, try just running it again. Sometimes it just fails. I don't know why. Just give it a second, try it again. And then maybe at least like one more time. And if it's still failing, then talk to me. Something else has gone wrong. Or maybe wait even just for a bit. Finally, we're getting to the step that is the actual problem. You can see I even checked for the version here. It doesn't work right now. I'm just going to remove that for now because it doesn't matter. I will probably add it in if I manage to fix it, but if not, then it doesn't matter right now. Frustrating. Okay, so what this is doing is it is downloading genomic information, clinical information, which it will use badly. And most importantly, the gene expression data. It is then storing all of that into the same object. And if that sounds complicated, it is. And you were right. It should sound complicated. It is. It requires like a multi-dimensional array. Let me pause the video real quick. I'm going to go in and look for I panicked for a second thinking I wasn't recording. I'm going to look for a diagram and just pull that up so I can show you a little bit what's happening. Okay, so the package that is organizing all this information into a single object here, described as SE for summarized experiments, it's called the summarized experiments package. It is a way to summarize biological data into a single object that allows us to combine genomic information, which is going to be stored in a section of the object called SE. So imagine that's like a container within a list type object. The row ranges is a function that allows us to extract that information, although I will point out the row ranges output is not a data frame and can't be treated like a data frame. And so oftentimes when I want to use the information in row ranges, I end up surrounding row ranges with a as.data frame, just to make it to something I know how to work with. Similarly, the clinical data, which is going to describe each of the samples that we're analyzing, is stored in a section called col data for column data, describing that this actually corresponds to the column of this middle section. Just as this was row data or row ranges, it described the information going across the rows of this object. However, in col data, what is the columns over here is actually described across the rows of col data. Kind of a weird transposition, but if you think about it, that kind of makes sense. Is if these were each of the different cases, here I have a data frame describing each of those cases as rows, but the columns are different descriptors, things like what tissue is it from, who is the hospital that submitted the sample, and things like that. Again, if I want to use that data, I typically use an as.data frame after the col data function. However, there is a handy tool where if you are using the $ tool, it actually does extract those columns from the sample data, not from here, which is really useful if what you're trying to do is understand your samples. So $ gives you kind of sneak peek access inside of col data, and it's very handy. In order to subset the clinical data, you can use code like this. This is, going to operate how we've been showing in the past. However, here is where you're using logic to subset the columns. The columns being these columns, not these. That's the most confusing part. In other words, if you want to remove samples, you use your logic in the column section of the extraction. If you are wanting to subset rows, you would use it in the row section. Although they also have some other tools They don't demonstrate it on here. My diagram, I think, is a little bit better, and it reminds you that if you wanted to subset the rows, you would just put logic to the left of the comma, because it would end up, it's organized based on this like central area. And so finally, let's talk about the central area. The central area is the central data. This is the gene expression data in R objects. So if I jump back to R, we can see what data managed to be downloaded. So if I take my DDS1 and I say assays, this is going to give me access to that center data. That's just from our diagram. I haven't loaded that library yet, so I can't do that. Okay, now that I've done that, when I run the code, it's telling me like, hey, look, you got these 6 different analyses with those different names. So if I wanted to, I could do unstranded. And this is just a big old data frame of our gene expression data. So this is legitimately the entire genome across each of those samples. This is the gene expression data. This is what we spend all this effort trying to get. It's organized into columns of samples and rows of genes. This is called the ensemble ID. I don't really like ensemble IDs, but if you're ever trying to figure out what those things mean, the row ranges, checking my spelling, DDS1. This is where you can find this information. So it does have like the gene name corresponding to these, and you can also look this up simply by going online and, just like searching for it. Like, what is this gene? And it's going to tell us about this gene, which is on chromosome 20 at position 50,934,852, and is named DPM1, which if it ever loaded, we would be able to see, but it's being really sluggish right now. So there you go. This one's working. So we can learn about DPM1. Cool. More on that later. Finally, I load the DESeq2 library. We'll come back to what that is, but it's kind of the thing that's doing all the heavy lifting as we get into the actual bioinformatics. For the next section, though, it's mostly just manipulating the data. Here is me viewing this data. Please remember that I said that the col data output's not a data frame. It's a little bit tricky to use, so I convert it to a data frame before then calling the view function. This allows me to visualize the data in a way that y'all are all familiar with at this point. I can scroll through here. One thing to note is that there's actually quite a few columns. Why does the R not show out of how many? So frustrating. I sure wish it did. But there is a lot of columns. Unfortunately, right now, some of these columns are wrong, and I haven't managed to fix it. Let me just make sure that's true. Yeah, it's still true. Like, I think this is, honestly, I think this is, so these are all colon samples, but I'm getting tissue from all over the body. I believe what's happening is that it's pulling these descriptors from other tissue samples of that same patient. But the data it's downloading is the data I actually requested, which is why I'm not really panicked. I'm only annoyed. But that's what's going on here. However, I do need to check a few things. We shouldn't have a duplicated sample submitter ID at this point, but just in case, I have this here. I even have some code to help you remove it. It is better to know which one you're removing, but sometimes it's just not worth the effort for like a small class like this, where this is not essential, since most of the duplicated values occur due to multiple samples of the same patient over a period of time, or simply because one round of sequencing didn't work well. You have to kind of dig into it to understand why. One of the things you can do that, though, is by taking out your duplicated sample. So I don't have any duplicated samples, but let's just pretend that this was the duplicated sample. And then you could go to the GDC portal. Oh, I'm glad you're working so fast today. So many updates. Anyways, and we can come in here and we can see, yeah, so despite it all, it was from a colon, despite it saying otherwise. And we can go in and we can see like, all right, well, what were all the files that were downloaded? This one, it looks like they only had, yeah, so they, yeah, there you go. They had 3 different samples taken. So this one was tumor primary tissue collected 2 years, 233 days ago. This one wasn't recorded. It was preserved using a fancy method of preservation, which sometimes causes problems, called fixed formalin preservation. I can't remember the E. Don't, okay. You should look it up, but I'm not gonna. And this is probably where I'm pulling that incorrect description from, even though I am in fact using the right value. Anyways, that's the kind of stuff that's going on. Cool. Next up, we've got some sections that are simply defining what these different terms are discussing. These different terms are the different sections. I did already cover that. If you're, ooh, that was bad. If you are coming here later, just scroll back till you see me going through a diagram that looks like, that looks like this. This is the diagram you'll need to see. It will be helpful. This is a weird object and it's like multi-dimensional and they're all interrelated. So go watch that section of the video, it's helpful. The extraction tools are a little bit complicated. I give you the documentation and then I also give you some tips in here on how to do it. The probably best tip I can give you though is just go on over to my diagram slide, which is under the project module, and I have several diagrams that show you how to use those tools to explore it. But here I am simply adding in some new clinical values. So as I said in that previous section, you can extract the clinical descriptors using the $sign. And that allows me to generate some comparisons, like are these samples in group 1 or in group 2? I can then store them in new descriptors that get added to the end of my DDS1's clinical data. So now, if I go over to here, and I go to the very end of the object, okay, it looks like I'm gonna have to, looks like I have to reopen it. It's not updated. There we go. Now, if I go to the end of the object, we can see it's a little bit longer. There's my new columns. Woo! Exciting. Okay. Once I've done that, I then take group 2, which right now is just logic, and I convert it into a factor. So we haven't talked a lot about factors, but factors are commonly used in statistical analyses in R because they are how we define a categorical value. So logic, not necessarily categorical. You could use it to describe, I guess, I mean, it's kind of categorical, right? But here, factor could have potentially included multiple values. But we're just doing very simple 2-level comparisons. And so this ends up being fairly simple code. But it could have been more complicated. But ultimately, this comp column is what's going to be used to control the experiment. I define that here, where I essentially say that the groups are as a function of, that's what the tilde stands for, the value stored inside of the comp column. It's got kind of weird syntax. If you've ever looked at the linear regression function in R, it has a similar syntax to there because that's where you're defining a relationship between things. Here, we're basically saying that the gene expression can be predicted as a function of comp, and that's what it's then going to test when it does the differential sequence analysis. When I run that, it kind of formats the data a little bit. That unstranded data set, which was our first assays value. So if I pull open the assays of DDS1, that assays has actually changed from unstranded to counts. And that's appropriate. The input to DESeq2 needs to be the raw count data. It doesn't use a common method of normalizing like the TPM calculation or the FPKM calculation. It does its own, a little bit more complicated method that's kind of outlier robust. It just handles outliers better, and it looks closer to like the medians of the different groupings. It's not exactly that. I honestly don't know all the details, but it's close enough that you can kind of think of it just as overall but robust expression across the different groupings. Here I'm just visualizing it again. It's going to look very similar, except now it's got a few more columns. There's that comp column, so we're looking good. Finally, I think this is finally, yeah. Finally, we are here just to help you understand your data. So like I am pulling out the project IDs. They should all be the same project for the vast majority of you. Here I'm pulling out the sample types. If you have a mixture of sample types, it depends on your project a little bit. Some projects, they do have multiple sample types. As a rule, like skin cancer just tends to have a bunch of different types of samples collected because we don't really know if it's the primary tumor oftentimes. And in those situations, you may just have to use different sample types. In general, though, you want to use the same sample type. Here, I'm demonstrating that, well, all of the controls I applied for pathologic stage aren't working. It's currently broken. Don't trust these values. Same for the tissue. Don't trust these values. You'll notice that a large number of them are colons because it's related to the right data. It's just pulling from some of the alternative values. It's quite frustrating. Hopefully I'll manage to fix that though later today. Okay, next up there is a PCA function. I don't ask you to know this deeply, but I'm going to dive into it just so that we can at least talk about what's going to happen. So inside of here it's just checking the inputs to make sure they are structured correctly. So the inputs to my PCA function, please note this is not your PCA function. You will be making a visualization of PCA function. I've done the PCA function though for you. It turns out it's not that hard. There's just some quirks of filtering that need to occur before we can run it. But the first thing I do is I check to make sure DESeq2 is present. I check the class of the input object, which I call DDS_fun. So it's instead of DDS1, which is what we're looking at, I renamed the input object to DDS_fun because it's the DDS version on the inside of the function. I check its class. Well, it needs to be this type of class. I check to make sure that there is a column called comp because, well, basically that's how I set all this up, is to be based on the comp column. So it needs to be present within the object. I then do an estimating of size factors. This is going to be part of the process DESeq2 does to calculate the normalization needed to compare different genes to each other and different samples to each other. So this is equivalent to like the TPM calculation, but this is just part of it. This is where it first generates, I think, the values describing the different samples. I have to go look at that. It might be the genes. Dang it, I haven't looked at this in a bit. One sec, let me pause and let me just go do that research real fast. Research concluded. It's both. It does both at the same time. So that's what I need to know. So it's very similar to the TPM calculation, as I said. Please note, it is uncommonly, for R, going to take the object that it started with. It's going to add extra information. Remember, this is a very complicated object with lots of layers, so it's going to actually add information inside of it. And so my output is then assigned to that original object, which is essentially modifying that original object to contain these new estimated normalized values. I then use the counts function that's also from DESeq2 along with the normalized equals true argument to extract those normalized counts from that object, convert it to a data frame, and store it inside of an object called norm counts. So in other words, I've calculated and extracted the normalized values. I then calculate the variance of the counts across each gene because only rows with more than 0 variance can be included in the next analysis. This actually is an essential step. It has to be greater than 0. I'm going to end up doing something that's not just greater than 0. I'm going to actually do greater than the median value. And this is partially just to make the analysis go faster and also focus the analysis on the most dramatic of the genes. This is actually an important step that you should be mentioning in your methods. So the first thing I do is I calculate the variance across each row in the norm counts object. We haven't talked about the apply function very much, but this function is equivalent to saying like something like for I in 1 through n row of norm counts and then taking the ith row, calling var on it, and I'll probably have to do unlist, and then assigning that to the ith position in per locus variance. So if I was to do that, I would have also had to have first established this. So I'd have had to say something like repeat NA in row times of norm counts. So yeah, that I believe would do it. Let's just try that real fast. Oh, I haven't run norm counts yet. Oh, yeah. To test this, I would have to run it. I just forgot I was in a function for a second. This is equivalent to this, or at least pretty close to equivalent to that. I might have made a typo is why I'm hedging. But this is much faster, right? So this is going to apply it across each row. Had that been 2, it would have applied it across each column. Had it been 1, 2, so like with a C function around it, it would have been across every single value in the data frame. Apply functions, very useful, much, much faster than loops. Anyways, that's what's happening here. In the end, that's going to result in a vector of the variance per row, which is each locus. I then say, all right, what is the median non-zero per locus variance? And then I say which of those variances are greater than that. So that's going to be approximately greater than the median variance, although it's technically the median non-zero variance. I then stop the code just in case you still didn't have any rows with variance, because that's what breaks this next step. Finally, I transpose the data. So I extract the rows that had a good PCA logic value. I then transpose them, making the rows samples and the columns variables. We haven't talked about this in class yet, but that's because that's the expected input for PRComp. PRComp is our principal component analysis. This is a function which is going to go each of those different variables, and it's going to try to reorganize the information in multi-dimensional space. Like I said, we'll talk about it later, in order to best spread out those rows of different individuals. This allows us to generate a plot as well as do some different calculations, which we'll talk about when we get to PCA. Finally, it takes that information and it plots out the appropriate sections in order to visualize the information. And it ultimately takes that data, stores it inside of a list object, and then returns it to the user. Stored inside of a function, this is what that looks like, run all together. So right now it has gone through, and maybe make my head get out of the way. Well, actually my head's not in the way. There's nothing underneath my head, just for the record. And that's funny. Okay, I'll just go away. Bye. Oh, I'll go over here. So these 5 samples really seem to be pretty different. You'll notice that this sample is sometimes up here or down here, and that's because the actual direction of this is basically arbitrary. This also could have been flipped left or right, depending upon the system. I did do some stuff to try to make sure that that didn't happen, but the actual algorithm doesn't care which direction it gets flipped. So with that, now visualize, I can start to make some decisions. I can say like, all right, I think that, let me move my head again. Sorry, that's probably nauseating. I think that, all right, bye. I think that the cutoff, like somewhere in this section, will be sufficient to help me isolate those. So I say here, I'm going to extract the ith section of PCA1 out. That was that list object created. If you weren't sure what's going on, you could just have visualized it. And you would have been like, okay, so, oh, okay, there's a section called PVE. We'll learn about, that's called percent variance explained. That's what's being used to generate these X and Y axis labels. But the first container, which is just simply named X, is going to contain the actual axis values. So for example, this PC1 and this PC2 is in $, square bracket, comma 1, and so this is my x-axis values, and this is my y-axis values. But I'm wanting to do logic based on this x-axis to remove these samples. And so I am saying, are those samples, gotta run that object first, are those samples greater than that cutoff value? For the few, or the 5, I guess, that are true, I'm going to end up wanting to remove those. So I save it to an object. I think I'm going to actually change this here. I'm going to say it's not good, and then not good, just to really have fun with y'all. So here, the PCA logic. It's true for everything except for the ones I don't want. Here, those bad samples are now being extracted. I always just like to make the thing I want as true. That's just my usual strategy for keeping organized. That's why I added those exclamation points. I think it does kind of make it a little bit harder to read, but y'all are smart. You'll get it. But I then use that to extract those values of the samples I don't want and store them inside of an object called bad samples. I then demonstrate how I can use that in an extraction to help me omit these samples I do not want. This is the syntax for removing samples from a DDS type object, a summarized experiment object. And one thing I want to point out is not all of your projects will end up doing this. So if you end up not wanting to remove anything, you could just add a comment here and create DDS2. What I don't recommend is commenting out this entire line of code, because then you're going to have to change everything later on because DDS2 no longer exists. I am going to run this code because I do want that subsetting to occur. And after doing that, I can now see that that went from 73 down to 68. Next up, I have the creation of PCA out 2, to out. This is where I am taking my DDS2 object, subjecting it to that principal component analysis again, which is going to help me see how do these rearrange now that these 5 samples are gone. So let's see what that looks like when it runs. It's going to take just a second. Okay, here we are, and we can see that the samples look much nicer if I flip back and forth between them. What used to be pretty spread out, there is now a sample over here, and remember the x-axis could have flipped and the y-axis could have flipped completely arbitrarily depending on your computer. But none of these look so far out that I think they bear removing. If I was going to remove any more, I would remove this one and probably none of the rest. at least on that first go, but I think this is sufficient. In your projects, you're going to come across the same issue. I do have a workshop day where we can talk to each other and just check to see, like, do I think, me, Nolan, if it needs to be removed. But for the most part, I just lean into whatever your TAs recommend. And so if they tell you to remove it, just remove it and move on with life. That's kind of true in a lot of these collaborative reviewing systems. Sometimes you just say, that's close enough, that this is a decision that is based on imperfect data. So like whether or not I should remove that is a decision based on kind of gut and instinct and experience. But I'm gonna say that's good, so I'm moving forward. And that moves us into the next filtering section. Now this filtering section, you probably want to listen pretty carefully because this is one of the ones that students tend to not really understand what's happening. Probably because there were 366 lines of code into this and you're getting tired. But let's try to understand it carefully. The first thing I'm going to do is I'm going to take that DDS2 object and I am going to use the counts function, but this time normalize is going to be set to false. This is going to give me the raw counts value out of the object. So note the raw, they're like decimal places and things like that. Had I said true, it would have looked like the, I have to run size factors. Let's not do it. It's going to take too long to do that. It would have been like decimal point values. But with the raw counts, they're whole numbers. Okay. I am then going to extract group 1. So if you think about that, you're not really going to be able to see it if I just print it. Instead, I can call dim on it. And I can see now it's just 19 columns. That's because that's how many samples are in group 1. I'm then going to compare that to the number 0. This is why we practiced all this order of operations. And right now I'm seeing mostly false, but every now and then I'm seeing a true. Okay. I'm then calculating the row means. So that means that trues are going to become 1s and zeros, or and falses are going to become 0s. And so if we scroll up to where we saw that series of trues, oh my gosh, it's so far up there. There it is. We can see that for that particular feature, every single sample in that group had a count of 0. And that's because that was a gene that simply wasn't observed. If we tried to follow up with it, we could probably learn a little bit about this gene. I don't actually know, so I'm just going in blind here. Oh, I must have copied the wrong code. There we go. Let's search that again. A protein encoded by the gene on a surface involved in leukocyte migration, T-cell adhesion, and probably a bunch of other things. There's even a post on what is this difference between the version that we're currently looking at with this par_y and the original version. This is, since by normal definitions there's no Y chromosome in female tissues, par_y quantifications should be ignored. Well, that would explain why it's not being added into there. Cool. Now I know. But anyways, we're going to store that information in this object, and then we're going to repeat it for group 2. So now that's an object, and that's an object. It describes the percentage of the row that had 0 counts per grouping. I then combine those values and I make a nice little histogram so that I can say like, oh yeah, we definitely got to remove all of those 100%. I do this really conservative approach where I remove any of them that are even close to 0. So I'm going to take 90%, which is probably around here, but we can try drawing that using the AB line function. Yep, that was about right. It's going to take all of these and it's going to remove them. To do that, I'm going to need to do some logic. So I'm going to take that object, I'm going to compare it to that cutoff established here. And then I'm going to say, is this true and is this true? That means that if this is true, it's going to keep it. And if it's not true, it's not going to keep it inside of this extraction. So if I run this, I saved it to DDS with no number because this is the one that I'm going to use in my final extraction. And I'm just double checking that my numbers are still good. And they are for my data. For your data, it's going to look different. But you can see the approximate range. In other words, we've really almost halved the total amount of genome. And that's because most genes aren't being expressed at any given moment in time. And sometimes there's even multiple versions of the same gene through situations like alternate splicing, which are absolutely not being expressed at all given points in time. So very tricky situation, but it's because not all tissues are doing the same thing. Different genes for different tissues. Many genes, though, for all tissues, but many also for specific tissues. All right, next up, we're just doing some checking, making sure that there's still some samples, or still some genes, still some samples. That's great. Class, this is checking the class of comp is still good. This is checking to make sure that you still have both values to be more than 6. We can see that all of those cases that got removed earlier were from my group 2, my group with the true value in comp. Next up, we've got call means. This needs to be 0 or above. Oh, sorry, this needs to be above 0. This is just calculating the raw number of counts per sample. Had this been too low, then we would maybe need to have removed those samples. That's not happening. This is checking to see how many in any single row have a 0 as the number the number of counts for that row and sample combination. And then it calculates the mean of that, which is going to tell you the proportion of samples across that gene that had 0 read depth. So it does that calculation, and then it calculates the max value. Since we removed earlier anything at 90% and more, we can see that this is really close to 90%, but it's less than 90%. And then it repeats that for group 2. In other words, it's just double checking our work, and we did a good job. And we are finally on group 6. Section 6. Section 6 is where the actual differential expression analysis occurs. I do have a link here to some more information on this package. There's a lot to learn, and I'll be honest, I don't know every single detail in this. I could teach a whole class on differential expression analysis, and I'll be honest, there's several workshops available where they do just that, and they are really good workshops, some taught by some professors I really respect and like. So I hope you take advantage of that if you are interested in this topic. But the first thing I'm doing is just to make sure that the object is still going to be interpretable and there's no issues. It's more of just a safety check to make sure you didn't skip around too much, to be honest. Finally, as we removed things, some of those normalization processes should and need to be updated. If I didn't include this, then it wouldn't necessarily update it after having removed those loci, and it would have been including those loci that probably didn't belong in there anymore. And so we're going to redo those size factors. And here it is. Here's the differential expression analysis. So far, all we have done is prepare and filter and reorganize and reorganize and filter and reorganize the data. But now is the differential expression analysis, and it's going to be a single line of code. Oh, there it is. There's not even a second argument. It's just DDS as an object. One of my favorite little quirks of this field is that that's often true, that most of my job is ultimately getting the data in the right shape for someone else to have gone through and built a whole package. So what it's doing is it first calculates those size factors if it didn't already exist, but ours did exist, which is why we needed to recalculate, because we had to kind of force it to happen. It then starts to look at the variance. I don't understand quite how dispersion differs from the concept of variance. I think it's just slightly different calculations, probably more complicated calculations if I'm being honest. It then starts to look at different features of the data, and if I try to define it, I'll probably say something wrong because this is not my expertise. But if you are interested in this, that's why you'll go and read that documentation that I pointed you to. But finally, now that it has a good feel of what the different groups should be for any given gene, it's going through and it's testing each gene to see, is there a significant difference in gene expression between the two different groupings. So it has finished that. It has found some issues with some of the genes. Sometimes it's going to just call the p-values for those missing and move on. We could dig into this for a while. I just want y'all to accept the default outputs. You know, again, if you are really digging into this, you would probably need to be trying some different models and making sure it's optimized for your system. But we're done. Note again, kind of weird, DESeq function actually saves the results to its input object. That doesn't change the object until we do an assignment, but that's exactly what I've done. I've taken the output, which is actually the whole DDS object again, but modified and assigned it back to DDS, overriding its old values and essentially saving the results of that experiment. However, those results are still stored inside of DDS, and so to actually pull them out, I use the results function, which pulls them out into an object that looks like this. So it's actually going to look like a data frame. It's slightly different. It is, I'm not actually sure what class it is. Oh, they called it a DESeq2 results object. I don't know why they felt the need to have their own class of object. I make it into a data frame. So here's me converting it to a data frame. I then bind it to a data frame made from the gene information just to make sure it's all nice and organized together. And altogether, I now have a big old data frame of everything I might be interested in. Finally, sometimes it causes problems if there are column names that are repeated. So for example, one of those column names is repeated. I could figure out what that is if I really wanted to. So I could say, all right, take those column names and extract the duplicated one. Base mean, it duplicated the base mean column. So I can't have duplicates. So instead, I just remove it. And we now have res output. That's it. Now I can do things like make a volcano plot. And here is a bare bones volcano plot with as simple as it gets methods. We can see my p-value here. This is a measurement of significance. And we can see my log 2-fold change. This is a measure of kind of the overall difference in average between the two groupings, defined as group 2 minus group 1, which means when group 2 is bigger, the value ends up on the right side, and when group 1 is bigger, it ends up on the left side at a negative value. This is just more to help you understand the kind of direction that is going between your two groupings, as well as see the overall difference, which is often, you're often more interested in more trusting of larger overall differences. On the other hand, the p-value is statistically based, and it gives us more information in terms of, like, should I trust this from a mathematical, statistical perspective? So anytime I say, is it differentially expressed, I am specifically referring to, like, is it significant in p-value? If I say, is it like strongly differentially expressed, that might be where I am saying, like, is it both significant, so high on this plot, and also interesting in terms of how far out it is and how strong that overall differential expression was. Last up, it has the save function, and this is going to save actually all of my objects to a single compressed object, which would allow me to, once it saves, and this does take a second, so I'm just going to pause while it does that. Okay, it's done. Now that that's done, I can do things like clear my entire environment. And you're like, no, I didn't mean to do that. Well, here's the nice thing is you can just run the load function and it is going to pull all that data back out of a saved object that had actually been stored on my computer, and it's able to recreate everything that we just did. Very handy, right? I use that all the time. There's a couple different versions. There's also, save RDS. Yeah, save RDS, which lets you save a single object. So I probably use that one more because oftentimes I just don't want to save everything because then I have to spend a lot of space and memory. And I often am wanting to more save like just the result of a single analysis. That said, that does work. Okay, so this is just a preview of what we're working on next week. This is where you will make a file, which I won't show here because this is the answer to the homework. But when you run this, it should load several functions into memory. It should load nice PCA, nice volcano, and res summary. These are functions that you will be writing. Nice PCA is not actually doing the PCA, although I guess it kind of is, because the PCA is actually when you use the principal component calculations. So it's actually kind of doing it, but that's not the point. It's not calculating the principal components. It's using your previously calculated principal components, using your grouping information, and making it into a nice GG plot. This is a visualization function, in other words. You got to make sure you structure your function correctly. That's where most students mess up, is they just get overwhelmed by my attempt to describe this, and they don't look closely enough at the usage examples to see that really I'm asking you to use this object and then like figure out how to write a function around that object. That's the real challenge. This is my first PCA. I had two, so I decided that I wanted to remove the samples, which right here you can see I've added a little line. Yours might not need a line if you're not removing anything. But after I decided to remove some samples, I then had recalculated my PCA. And so I visualized that nicer PCA again, and so I can see it right here. I also have a nice volcano plot. This one takes a second to plot. It's a pretty complicated volcano plot. But this function allows me to make nice little visualizations. I can even do things like adjust some of these parameters if I want to, and have more stringent cutoffs if I feel like I need those for some reason. Now, for the record, you shouldn't do things like arbitrarily pick the cutoff. I really only want to see you either use 0.01 or 0.05 because those are pretty much the standards for significance in this field. That's going to be 5 out of 100 times. I don't know if I trust the value, or 1 out of 100 times I don't trust the value. In the end, though, these are not you making medical decisions with these plots. This is you exploring correlations. And so the values should reflect really what's a realistic acceptable error rate. Well, for an exploratory study, I'm okay with a 5% chance that the gene I've picked is unrealistic. Now, for the record, I'm doing lots of tests, so that number gets a little bit hard to interpret. But that's why I'm using what's called the adjusted p-values and not just the regular old p-values. That helps to adjust for the multiple experiments, because since each of these different genes represents an independent-ish sampling of distribution. The 1.5 here is my log2 fold change cutoff. That one actually is pretty arbitrary, but again, you shouldn't base things on simply what makes a pretty plot. You should base it on what is like the kind of norm for the field. And the norm for a lot of these fields is somewhere between like 1 and 2 and maybe even up to 2.5. I've cut the middle here. I've said 1.5. At 1.5, we're typically in pretty big changes overall between the groupings, and these are often going to be of like interest to someone studying and wanting to follow up on any of the genes we find to be differentially expressed. Finally, my plot isolated the genes in this corner and in this corner, and it plotted the names of, I think, the top 5 genes per corner. There's a lot of ways that I accept that you do the names. Really what matters is that it works for your plot. This is where what matters is like what makes a nice plot, because really the only ones that should be labeled or somehow indicated are any that you are then talking about in your analysis. So here I've been pretty safe, and since I'm probably going to be talking about these two, I labeled them. But there was plenty of room on the plot, so I also labeled these and these over here. Finally, res summary just produces a series of printed in message that describe what's going on. And this is why I said, like, your results are almost going to be just copy paste from your function. But there you go. This is where you should then just reminding you to go back and make sure that you change all of these little Q numbers into the actual comments as part of the annotation study. So well, I hope that helped. 1 hour, 25 minutes, about what I expected. Thank you, and I'll see you tomorrow. Bye.